So we've seen how the muscle is able to be recruited, but now we have to figure out, okay, how can we go ahead and take that recruitment and start developing the actual strength of the contraction? And this is where we get into the sliding filament theory. The sliding filament theory explains how the myofibrils are able to generate force and how that force being generated increases the strength within the muscle to allow for the contraction to take place. In this, we're going to look at the roles of actin and myosin within each other and around each other. So we have to remember that myosin is going to consist of two twisted strands of globular cross-bridge proteins that are going to project towards the outer strands of actin, where actin is this chain of globular proteins that have distinct binding sites that are set for the globular cross-bridge heads of the myosin. Regulating the contractions are two proteins and an enzyme. The two proteins are troponin and tropomyosin, which are going to regulate the binding site on the actin, and the enzyme is the myosin ATPase. And so when we look at the overall structure of what we're looking at our globular chained actin molecules with the troponin and tropomyosin regulating the ability for the myosin head cross-bridge to link onto the active site on the actin molecule. With troponin, we have different types of troponin, and troponin itself is made up of distinct subunits. Of the different types of muscle fibers within the twitch characteristics, the difference is going to exist in what's referred to as the troponin C, and that is how affinity does that troponin have to calcium. That is how quickly can calcium bind onto it. And then the troponin T part, which is how quickly can it do a change of configuration based off of calcium binding onto the troponin C part of the molecule. Sitting at the base of the hinge part of the globular heads of the myosin is a ATPase, which is going to hydrolase ATP to allow for the power stroke to take place. And so let's take a look at this in schematics, and we'll take a look at it in terms of an animation. So what ends up happening is we can think of this as a four-stage process in which calcium will bind onto the troponin, causing the conformational change, opening up the binding sites. Once the binding site is opened, we will hydrolase ATP into ADP and inorganic phosphate. This is going to lead to the linkage of the myosin head to the actin binding site. When that linkage takes place, it causes a dissociation of the ADP. The dissociation of the ADP causes the myosin head to do a conformational change, opening up the binding site for ATP again. In that conformational change swing, it pulls the actin towards the M line. We then get into the resting phase, and the cycle will continue as long as we have calcium being released into the sarcomere and ATP being regenerated to allow for the contraction cycle to take place. So what is this going to look like? So what ends up happening is we have calcium coming in and causing the ability to have the myosin heads start interacting with the actin via the conformational change. Notice the pathway of contraction. Now notice we're showing this only on one of the strands of actin, and this is going to be in a 360-degree cogwheel within the cylindrical myofibril. So let's go ahead and put this all together. Let's put together the excitation contraction with the sliding filament, and let's look at it in terms of a schematic feedback loop here. So we have the motor neuron coming down. It's going to release acetylcholine, which is going to lead to excitation at the neuromuscular junction. If we reach threshold, calcium will be released from the sarcoplasmic reticulum, which is going to lead to binding of calcium to the troponin, which is going to allow for the conformational change and the tropomyosin revealing the actin binding site. We now need two things. We now need ATP and we need that calcium to stay there. So ATP is going to hydrolase and allow for the myosin head to do its conformational change, which is going to allow for the power stroke to take place within the myosin head as the ADP and inorganic phosphate unbind. As long as we have another ATP available, the myosin head will go about and connect with the actin molecule, as long as we have calcium that is still available to allow for the binding site to remain open. And so we have this cycle that will continue as long as ATP is available for the conformational change to take place, and calcium is present within the sarcomere to allow for the troponin to do a conformational change to open up the binding site by pulling away the tropomyosin. We're going to take a look at factors that will impact this when we talk about fatigue, but here is the general cycle of what's going on.